My name is Nyla Crayton. Welcome to Unit 3 of AP Biology. Today we'll be teaching about the process of photosynthesis.
Photosynthesis is a process by which glucose is made by plants in order to provide energy. The glucose is then transported to the cytoplasm for glycolysis, and if the organism is capable, to the mitochondria for aerobic respiration.
Photosynthesis is an endergonic reaction, meaning that the process requires energy for it to work. This differs from cellular respiration in the sense that this process requires energy, whereas cellular respiration releases energy. Also, the reactants and products are almost exact opposites, whereas cellular respiration takes in glucose and oxygen and releases carbon dioxide and water and energy. Photosynthesis takes in energy, water, and carbon dioxide and releases oxygen and glucose.
Photosynthesis consists of two reactions, with each reaction taking place in a different part of the plant cell's chloroplast. These reactions are the light-dependent and light-independent reactions. Respectively, these reactions take place in the thylakoids of the chloroplast and the stroma of the chloroplast.
While the reactions and products of cellular respiration and photosynthesis are completely different, some of the basic processes are the same, and some might even be familiar. Take a look at our images. As I mentioned earlier, these are chloroplasts and this is the mitochondria. The different parts are the outer membrane, as you can see, inner membrane, thylakoids, stroma, and gramma. This is where the reactions take place, such as the Calvin cycle, light-dependent, independent reactions, along with like chemiosmosis, the electron transport chain, etc. The mitochondria mainly occurs in animals, except for it occurs in all eukaryotes. It has a mitochondrial matrix, which is where some parts of the cycle takes place, the inner membrane, and then the outer membrane.
The light-dependent reaction takes place in the thylakoid membrane. The reaction is initiated when a photon of light reaches photosynthetic pigments through the leaf, which is where all light is received. When the light reaches the leaf, hydrogen is broken apart from oxygen and water, and oxygen is released.
After this occurs, the energy is transferred through the light harvesting complexes of photosystem 2 to the reaction center complex to the primary electron acceptor once it reaches a certain energy level. This is called a redox reaction. Then it is passed through photosystem 1 via an electron transport chain that produces ATP. This is because as electrons fall to a lower energy level, it releases energy.
ATP is synthesized as the electrons pass through the cytochrome complex, as you can see, building a proton gradient. Then the reaction is passed on through photosystem 2, where NADPH is produced, and will power the reactions in the Calvin cycle. This process converts light energy to chemical energy, which will ultimately also be used for the synthesis of sugar in the Calvin cycle. The following image shows light-dependent reactions. This is when concentrations of H+ ions go from a high concentration to a low concentration. It goes through photosystem 2, the cytochrome complex, photosystem 1, and then it goes back down through ATP synthase, which produces ATP from ADP plus a phosphate ion. This is going through the thylakoid membrane. It needs these inner membrane proteins because it uses active transport and it can't go by itself. And the thylakoid membrane is made up of semi-permeable phospholipiid bilayer. It only lets in small nonpolar molecules, not nice big polar molecules such as H+ ions. As you can see, it is ATP synthase that actually produces the ATP, and electron carriers from other cycles, such as the Calvin cycle, actually move the H+ ions through the proteins, which is what allows a proton gradient to form, which produces the ATP.
The Calvin cycle utilizes carbon dioxide along with the NADPH and ATP from the light-dependent reactions in order to make G3P from a process of enzymes and intermediate molecules. This Calvin cycle produces glucose, which is then transported to the mitochondria for cellular respiration. Photosynthesis can be in multiple different forms, such as C3, C4, or CAM. Each one has its own process of photosynthesis and contributes to the ultimate biodiversity of the plant kingdom.
The molecular level process of the Calvin cycle begins when carbon dioxide from the atmosphere enters the chloroplast's stroma. There, an enzyme called rubisco anabolizes the carbon dioxide with a molecule called ribulose bisphosphate. This creates a short-lived intermediate molecule that soon breaks down into 3-phosphoglycerate. An ATP molecule then phosphorylates the 3-phosphoglycerate to make a 3-bisphosphoglycerate molecule. Then, an NADPH molecule that was produced in the light-dependent reaction is oxidized to turn the 3-bisphosphoglycerate into 6 G3P molecules, one of which exits the chloroplast and binds with another 3-carbon sugar to become glucose for respiration. The remaining 5 G3P molecules are then recombined and phosphorylated by another ATP molecule to become more ribulose bisphosphate, and the cycle starts over again once more carbon dioxide enters the chloroplast's stroma.
C3 plants. This uses the enzyme rubisco. In C3 plants, the plant cannot close the stomata when it is hot and dry. Also, the enzyme rubisco has the same affinity for O2, which can leak out of the thylakoid membrane, and CO2, which is normally used for the Calvin cycle. The problem is it photorespirates instead of using the Calvin cycle, while the Calvin cycle actually is needed to reduce G3P, which is used in other processes. This is why C3 plants are worse than C4 plants.
C4 plants always use the Calvin cycle, but instead use PEP carboxylase instead of rubisco. C4 plants can close their stomata when it's hot and dry to produce more oxygen and ATP. And PEP carboxylase does not have the same affinity for oxygen and carbon dioxide, meaning it only takes in carbon dioxide. But they do use rubisco to reassimilate the carbon dioxide release into organic materials.
Cam plants are water-storing plants that are found in arid conditions. Some examples are pineapples and cacti. What makes them special is that they open the stomata during the night and close them during the day. Doing this helps the plant store water and keep CO2 from entering the leaves. The CO2 that is taken in during the night helps produce organic acids. That process is called the crassulation acid metabolism. The mesophyll cells store organic acids in cam plants. CO2 is released during the day when the light reactions can make ATP and NADPH. The difference between cam plants and C4 plants is that the two steps occur at different times, but in cam plants they happen in the same cell.